have class in a building like this when it's so nice outside? No, I'm not either. All right, so a couple things. One, I keep forgetting to bring note cards. You will get note cards for the exam. You will, like last term, you'll need to get them from me. And if somebody is really nice and reminds me to bring them on Monday, I will bring them on Monday. So you can have them to work on, okay? Somebody reminded me actually on Wednesday, but I didn't bring them today. In any event, so note cards. As I said last term, you have to get them from me, and you have to turn one in whether you use it or not. So make sure that you get a note card from me. Okay, we're moving our way through gene expression, and we talk about a couple things in prokaryotic gene expression. I'm going to finish those up, and then say a few things about eukaryotic gene expression, and get you hopefully out of here in a reasonable fashion today. So we'll try not to do too many things. When I finished last time, I was at about this point, and I showed you what happens with bacterial cells when they encounter lactose. And the point of this slide was to simply show you a phenomenon known as induction. Okay? When I was your age, induction meant something very different than it means with what you see on the screen. Induction meant you were getting drafted to go to a war in Vietnam. And so this is a much better kind of induction. Induction happens when some cells receive some signal of some sort that make them do something. In this case, what's happening is this enzyme to break down lactose is being induced. It's being synthesized where it wasn't being synthesized before. And the signal turned out to be coming from lactose itself. So when lactose appeared in this cell's food, then the enzyme to break it down was synthesized. How does it work? All right. Well, there's really only one enzyme the cell needs to break down lactose. Turns out that there's an Operon that has 3 genes, and I'll say a little bit about those in just a bit. But there's only one of those 3 that's needed to break down lactose. And it's an enzyme called beta-galactosidase that you can see right here. Lactose, you recall, is a disaccharide. It has 2 sugar residues in it, 1 glucose and 1 galactose. And so in order to metabolize lactose, cells have to break it down. And then you remember glucose, of course, can go through glycolysis, and galactose can get converted into intermediates in glycolysis so that it can get broken down as well, but this has to happen. So cells don't want to make beta-galactosidase if there's no lactose, because that's a waste of energy, and they want to make a lot of beta-galactosidase when there is lactose present. So we have to understand how it is that they do that. Okay? Before I tell you about how they do it, I want to tell you something about a very cool trick that biochemists came up with. I mentioned last term that biochemists are basically lazy people, and they like to find the easiest ways that they can to measure something. And we talked about this with respect to serine proteases, where we saw a serine protease could cleave an artificial substrate that gave a yellow color. And so we could see how much of the enzyme was actually working by measuring how much yellow color was produced. Well, similar things have been created for studying beta-galactosidase. And what you see on the screen is a molecule that we call XGAL. And it looks enough like lactose to fool beta-galactosidase. So beta-galactosidase, when it encounters XGAL, will treat it as if it were XGAL. And it will cleave the bond between this guy right here and this red residue on the side. Okay? Well, the upshot of that is if this cleavage occurs, a molecule with a blue color will be produced. Okay? And so like we did with the serine proteases, we can measure the amount of beta-galactosidase present by simply measuring how much blue color is present. And that turns out to be a very, very useful tool, as we shall see. So beta-galactosidase can be measured very easily in that way. I said a little bit about operons, and now I want to show you up close and personal the beta-galactosidase operon. So we're looking at a segment here of the E. coli chromosome, and in this segment of the E. coli chromosome, we have several things that are important. I want to emphasize that the operon itself is located from here to the right. That is from the purple, well, actually from here, the purple to the right. Okay, and this is a blow-up of this region here. But from this guy to the right, or in essence, from this to the right. Okay, so the operon itself, all right, is a coding region. It codes for 3 different genes. Why do I show you this guy over here? All right, this purple region. This purple region on the left is not part of the operon, but it codes for a gene that helps to regulate that operon. Okay? Now, what's the difference between being over here and being over here? If you recall my definition of an operon, I said that an operon is a collection of genes that are all controlled by a single promoter. The promoter is right here, and so there's no way that this promoter can make that gene. So this gene over here called I is not a part of the operon, but I encodes something very important. In fact, what it encodes is the Lac Repressor. So the Lac Repressor is, its coding region is right next to the Lac Operon. How convenient, right? And the operon is over here. Well, for our purposes, excuse me, we're very interested in this gene right here, LAC-Z, because Z is the gene that codes for the beta-galactosidase. There are 2 other genes that are present, and for our purposes, they don't play a very important role, but I'll tell you what they are. One is a protein called a permease. And the permease allows the cell to more efficiently absorb lactose. Allows the cell to more efficiently absorb lactose. So when the cell detects that there's lactose there, it makes permease, which lets even more lactose in, and it makes beta-galactosidase so that it can break it down. Okay? Last, the cell has an odd enzyme called a transacetylase, which you don't even need to know the name of. And the transacetylase modifies lactose slightly. And when it does, it creates a byproduct called allolactose. Now, this gets a little confusing. And for our purposes, we will assume that this allolactose is the same as lactose. For our purposes, we're going to assume that. Okay? And if you're curious about why I make that assumption, or curious about how this actually occurs, I'd be happy to tell you separately. But to keep things simple, we're going to treat allolactose as if it's lactose. All right. So these are the 3 genes of the Lac Operon. Excuse me. You'll notice that there are control sites, and the control sites are actually enlarged a little bit, so we can see down here. We see P and O, and your book is using that to stand for promoter and operator, although I tend to say that the operator is really part of the promoter. Okay. So promoter and operator. The operator is that region that you think about where the Lac Repressor binds. How does this operon get regulated? Well, it turns out to be fairly simple. Yes? Yes. Okay. How does this operon get regulated? Well, it starts out with a negative regulation. And negative regulation means that Lac Repressor, when it's present in the cell, and there's no lactose present, will bind to the operator. The Lac Repressor binds to the operator. RNA polymerase cannot bind, and as I said last time, transcription is completely turned off. There's no synthesis of this operon. Okay? However, when the cell encounters some lactose, lactose in a modified form gets bound to the repressor. The repressor can not only bind to the operator, it can also bind to a modified form of lactose, and we're going to treat it again as if it's just simply lactose. What happens? Well, it can't bind to both. When the repressor binds to lactose, it lets go of the operator. And this is a signal to the cell, the promoter is wide open. Now we can start transcribing the operon, and we only transcribe, or the cells only transcribe the operon when lactose is present. If lactose is absent, the repressor turns it off. If lactose is present, the repressor is gone, and RNA polymerase jumps in and starts making the operon. It's a very simple but very, very useful system. Okay? One molecule, bang. The cell detects it's there. Oh, it's time to start making enzyme. Very, very cool. Very simple. Okay. Now, there's the Lac repressor bound to DNA. This is actually the allolactose I talked about. This is actually the molecule that binds to the repressor. We're not going to focus on it. We'll treat it as if it's lactose. So don't sweat. How do you get lactose if you don't have the operon going? All right. Last, I need to tell you about this guy here. Okay. So let's say I'm a researcher, and I want to study the way in which the Lac Operon works. All right. I want to study the way in which the Lac Operon works, and I've got a bit of a problem. I give cells lactose. What's going to happen? Well, those cells the lactose is going to bind to the repressor. It's going to start synthesizing the operon, and what's going to happen to the concentration of lactose over time? It's going to fall. So I've got two variables in my equation. I want to study this operon so that I see what happens when lactose is present, but then lactose itself starts disappearing. I've got a problem. It'd be nice to be able to find something that binds to the repressor that doesn't get broken down. So I can see what happens with this over time. And so that's what this molecule is. It's called Iptg, isopropyl thiogalactoside. I never can remember that myself, so you'll probably call it Iptg like I do. Iptg has the very useful property that it will bind to the Lac repressor just as if it's lactose. But, importantly, the beta-galactosidase won't break this down. So once I give this to induce the operon just stays on. Okay? There's no running out of the lactose material that would be needed to bind to the Lac repressor. So now I've got an inducing molecule. It's artificial. It's man-made. It's not a natural product of any sort. It's a man-made thing. But now I've got something that I can turn on the operon and it won't itself get broken down. That's really useful. So two things I've given you are artificial, xgal and Iptg. Iptg is an artificial inducer. Xgal is an artificial substrate. Xgal gives me a blue color. This guy is going to give me beta-galactosidase. We're clear? Now this turns out to be really useful for biotechnology purposes. Let's say, for example, that I have a gene for human growth hormone, and I want to put it into bacterial cells, and I want the bacterial cells to make a lot of human growth hormone, and I want to control it. I'm a control freak, right? So what do? I take the Lac promoter, I throw away all the operon, and I put my gene in front of the Lac promoter and the Lac operator. Okay? I let these cells grow up, and what do? I treat them with Iptg, and what's going to happen? They're going to start transcribing my gene like crazy, and they can't break down the Iptg. They're going to make a lot of messenger RNA for my gene. They're going to start synthesizing my gene and make me a very cool product. So this turns out to be very useful for biotechnology purposes. Okay. Yes?
So will Iptg bind more than just the Lac Operon?
Will Iptg bind more than the Lac repressor, I think is what you're asking. And in general, no. No.
So you have to have a different one for human growth hormone?
Well, all that you care about is that you're making the messenger RNA for human growth hormone, right? So if you put the human growth hormone in front of the promoter, then this is turning on the promoter. You're making messenger RNA for human growth hormone. That's going to be translated into human growth hormone. And the aim here is you're making a bunch of RNA. Make sense?
Yeah.
Yes, sir.
Wouldn't you have to at the same time engineer some export properties into the cell or lyse them all at once to harvest the properties?
Yeah, so there's other considerations. Would you have to export it from the cell? Would you have to bust the cells open? There's a lot of other considerations in terms of getting your final product. But this is a way to make your final product that's going to be useful to you. Yeah. Okay. Well, let's see. This is simply reminding us that when, here's the Lac repressor, shown in purple. When there's no lactose present, the repressor binds to the operator, the operon's turned off. When lactose is present, this inducer molecule binds to it. This repressor can no longer bind to the operator. The RNA polymerase comes in and does its thing. Make a lot of beta-galactosidase, permease, and the transacetylase. Okay. Interestingly, if we look across the entire E. coli genome of 6 million base pairs, that's million, not billion, 6 million base pairs, okay? And we look for, are there other things that use the lac promoter, for example, things that might be turned on when lactose is present, we see that there's nothing else. The only place we see the lac promoter in the E. coli chromosome is where the Lac Operon is. We don't see it for other things. Okay, it's only there. Now, that's not true for all promoters. Here's a promoter that is necessary for making purines. That's why it's called the PUR operator. We see that promoter appearing in a variety of places. Why would we see one promoter appearing in a variety of places and another promoter only appearing in one place? The answer is the need to coordinate things. Okay? So if I'm going to make purines, it might take a bunch of different genes in different places on the chromosome to be made. So let's imagine, you saw when we went through the nucleotide metabolism, you saw it took about 10 genes. It took a lot of things to make purines. We might imagine that, well, here's a few of them over here, here's a few of them over here, here's a few of them over here. Once I have the signal that says, hey, it's time to make purines, all of these different operons get turned on. Okay? Whereas in the case of lactose metabolism, the only thing we really need for lactose metabolism is beta-galactosidase. We don't need anything else. It works on one operon and we've got it. Okay. Well, there's yet another piece to this puzzle of the Lac Operon. It's interesting. Okay? It's interesting. The promoter of the Lac Operon is not, underlying not, an ideal promoter. We talked about the Tata box and how it was, if the closer the sequence was to that TAT, the stronger the promoter was. Well, if you look at the Lac promoter, it's not very strong. It doesn't look very much like TAT. So if we rely simply on that promoter sequence and nothing else, we would get some synthesis of messenger RNA, but not an awful lot. Okay? Well, it turns out that there's something else in the Lac promoter region that really turns on the transcription. Okay? And what it is a binding site for a protein called CAP, C-A-P. Okay? CAP is also in some places called CRP. And basically what it is a protein that, when it binds to cyclic AMP, binds to this region next to the Lac promoter. You see it depicted here. And what it does is these CAP or CRP proteins favor the binding of RNA polymerase. They act like a super sigma factor. They really help recruit and get RNA polymerase to come in there and start transcribing messenger RNA. So because of CAP, the Lac promoter turns out to be very strong. Does CAP have the cyclic AMP play a role in this process? It does. Okay. Is cyclic AMP present when cells have low energy? And the answer is, to some extent, yes, it is. Now the next question is, if I have CAP binding and I have the Lac repressor binding, who wins? It turns out they can both bind. And the answer is the Lac repressor wins. Because as much as this would try to load it on, if the repressor is sitting right here, there's no place for the polymerase. So most of the time, the cell, or I shouldn't say most of the time, but a good deal of the time, the cell actually has CAP proteins sitting there waiting for things to happen. But if you think about it, it makes sense. The cell doesn't want to transcribe this operon unless lactose is present, and the repressor won't leave until lactose is present. Very simple, very cool system. Now, I'll stop there and take a question, since there's usually questions about that. Yes, sir?
So CAP binds to the promoter?
Cap binds to the region of the promoter. That's correct. Pretty cool. Orienting yourself, cap is up here, RNA polymerase is here, and the Lac Repressor binding site's about right here. That's the physical orientation of them on the chromosome. And of course, the gene's going to be down here. The operon's going to be down here. Okay? Good. Cool. All right. That's the binding. Not surprisingly, cap binding induces a bend in the DNA. And you've seen the role of bends in DNA in terms of opening up strands. That's going to be important for RNA polymerase, and it's one of the ways that cap binding facilitates the binding of RNA polymerase. Okay, so that's what I want to say about the Lac Operon. I'll actually come back and say a little bit about it later with respect to eukaryotes, interestingly enough, more as a tool than as a phenomenon in eukaryotes. Okay. Now, I want to spend just a minute, and I'm not going to spend much time on this because I think it's, to be honest with you, a bit overrated, but I want you to have an idea of other types of regulation that bacterial cells have. Okay? Bacterial cells regulation is relatively simple, and this one you'll see is, oh, it's not very simple, but it actually is fairly simple in terms of structure and requirements. But I want to show you something because it tells you something very cool about amino acids in cells. What I'm showing you on the screen is a depiction of the 5' end of the messenger RNA of the tryptophan operon. So the tryptophan operon is a very long sequence of RNA that gets made. It's about 10 different genes that are in this operon. So it's a pretty long messenger RNA. And the operon is involved in the proteins, I should say the proteins that are coded by this operon are involved in helping the cell to make tryptophan. So the cells have to make a decision. Do I need to make tryptophan or not make tryptophan? Because I don't want to be making this very long messenger RNA unless I really need tryptophan because, I'm going to be wasting energy if I'm not careful. All right? Well, the way that the cells make this decision is by this phenomenon I'm getting ready to describe to you called attenuation. Okay? Now, your book doesn't have the best figures for this. And it shows you two possible structures that can exist in this region. Okay? So let me show you what we're looking at here. All right? We have I said there's 10 different genes that are in this messenger RNA. Okay? Very close to the 5' end is a little tiny, look how short that polypeptide is. There's a little tiny coding region for a polypeptide. And 2 of the amino acids in there specify tryptophan. These 2 amino acids the cells use as a barometer to determine how much tryptophan is present in the cell. Okay? You also see 2 regions further downstream, one that's shown in red and one that's shown in blue. These are called the region of the attenuator. Now, what I don't like about what your book does is it doesn't show the most important thing, which is that a portion of the red over here on the right can actually interact with a portion of this blue. So we really have 3 possible structures. This one, or a sort of a hybrid between the 2. That hybrid between the 2 turns out to be important. Okay. How does this work? Well, let's imagine that the cell is in a situation where it has plenty of tryptophan. It doesn't want to be making this operon, but the control of this expression of these genes is not at the level of transcription, at least not at the level of initiation of transcription. Rather, it's at the level of termination of transcription. So the question that the cell has is, it's always going to initiate it. The question is it going to terminate it early, or is it going to terminate it only after all the genes have been copied? Okay? It's going to want to terminate it early if there's plenty of tryptophan, and it's not going to want to terminate it until all the genes have been copied unless there's low tryptophan. Right? What you see on the screen is a depiction of what happens when there is plenty of tryptophan. What happens? Well, RNA polymerase starts making the messenger RNA, and it's gone, it's made all this stuff up here. And you recall that we're in prokaryotes, so once we've started getting to coding regions, the ribosome can come along and start translating behind there. Okay? So the ribosome is following the RNA polymerase along, doing its thing. And what's happened is if there's plenty of tryptophan, the ribosome just goes scooting through this little region. Remember, there's 2 tryptophans in this little peptide. It doesn't get slowed down waiting for tryptophan because there's plenty of tryptophan, so the ribosome comes scooting through. And when the ribosome comes scooting through, it encounters this red region. Okay? And the red region, when it's covered up with the ribosome, can't do that sort of red-blue pairing that I talked about. And instead, the blue pairs with itself. Well, the blue, guess what, is a terminator. We talked about those factor-independent sequences that can lift the butt end of the polymerase up and kick the polymerase off, and everything falls apart. Look what happens. When there's plenty of tryptophan, termination happens early, and we only make this one little tiny sequence. We don't make all the others. The cell doesn't waste too much energy. Now, let's imagine what happens when there's very little tryptophan in the cell. When there's little tryptophan, the ribosome starts doing its thing, and it gets to that region where there's 2 tryptophans, and it says, where's the tryptophan? Where's the tryptophan? Where's the tryptophan? It's sitting there waiting. The tryptophan concentration is low. And remember, we're talking about a diffusion process to get those tRNAs in there. So if tryptophan's low, the ribosome is going to sit here and wait for a while for the transfer RNA with the tryptophan on it to diffuse into the A site. Because the ribosome is slowed, what happens? Okay? Well, what happens is basically, and this is a little inaccurate, what basically happens is this red guy pairs with this blue guy and stops the blue guy from being a terminator. In the first case, we had covered the red guy up with the ribosome. In this case, the red guy is not covered up. So we can think of the red guy as an anti-terminator. When the red guy forms like this, or the red guy forms the red-blue complex, there's no termination. Now RNA polymerase says, okay. And now it goes along, merrily along its way, and makes the entire operon. So what I've just told you is that when there's low tryptophan, there's no termination. When there's abundant tryptophan, there's early termination. Okay, I'll stop there and take questions. You guys are really quiet today. Yes, Karen.
I don't really understand the process behind how that terminator blue region
Yep.
forms.
Okay, good question. So she doesn't understand, when does it make a blue duplex and when does it not make a blue duplex? The answer to that question is based on the position of the ribosome. Okay? And that's why the red sequence is important. That's why I don't like this figure. Okay? Let's go back to the previous figure. Previous figure, the ribosome is moving merrily along because there's abundant tryptophan. It doesn't get stuck over here. It goes and it covers up part of the red sequence, the anti-terminator. The anti-terminator can't stop the blue from forming. Okay? As a consequence of that, the blue forms, termination happens. On the other hand, when the ribosome gets stopped, right here, there's nothing to stop this anti-terminator from anti-terminating, as it were. Okay? As I said, in reality, the red is pairing with part of the blue, and when the red's pairing with part of the blue can't pair with itself. That means then that no termination happens when there's low tryptophan. Does that help? A little.
It's more like the mechanism behind what causes it to hold in on itself. Are they just like complementary?
Okay. Very good question. So what caused it to fold in on itself? If the pairs, if the base is there can, they will. The sequence is always the same. So the question is the blue free to itself only, or is it having to interact with this red guy? And so the ribosome really determines whether or not the red guy is available to block that blue from pairing with itself. Does that make sense? Other questions? Jody.
So that little chunk out front, was that just the little short 10 or 15 chunk we saw, or is there like 1 or 2 genes that it does get through copying?
No, his question is, are there other things up there besides that 10 or 15 amino acid sequence? That's it. So there's 10 or 15, there's a little space, then there's another gene, another gene, another gene, a whole bunch down the stream. So we're looking at a short region of non-coding between this first little segment and the next gene. It's a very cool system. Now, it's not just an oddball system. It turns out that several amino acid operons in E. coli are regulated in exactly the same way. This shows the operon region for several. Right? You could probably look at this and tell me which operon is involved in A. I hope. Especially since it's in red up there. Okay? Look at the threonines. All right? Why do we have all these? Why does the cell have all those threonines there? Well, if threonine's in low concentration, the ribosome is going to slow down going through here. It's going to let that red region interact with the blue, and no termination will happen. The operon will be made. So as a consequence, when threonine is low, the cell is going to make the threonine operon. When phenylalanine is low, the cell is going to make the phenylalanine operon. When histidine is low, the cell is going to make the histidine operon. Simple system. Very, very efficient at helping the cell to make the right amount of a given protein. Yes, sir?
Is there any particular reason why it's all amino acids that have relatively large side chains?
No. There are some valine, for example, is also in this category. So it's not just coincidence that these have large side chains. No. Okay. Let's see. You guys want to sing a song? Okay. So I actually have 2 songs today. If we get far enough, we'll do a second one. The first one is, let's see. Yeah, this is the first one here. The first one is one that some of you may know. I don't know, but it may not be a big one. The song Maria from West Side Story? Yeah. Okay. So this one's a little theatrical, so we'll have to get silly. Translation. The most intricate thing I ever saw. Here's where you come in. From 5' to 3', translation, translation. The final step that we know about the central dogma. Amino, carboxyl, translation, 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 translation. I just learned the steps of translation and all the things they say about tRNA are true. Translation, to form peptide bonds in translation, the ribosomal cleft must bind to ADF TU. Translation, AUG binds the F-MET's cargo. 16S lines up Shine and Delgarno. Translation, I'll never stop needing translation. The most intricate thing I ever saw. Translation. Okay. Thank you. Okay. What's that? I always start too high. That's my problem. All right. If we get far enough, we'll have another one before the end of the period. All right. Let's talk about gene expression in eukaryotic cells. And in eukaryotic cells, I hope by the time I'm done with this, and I'm not going to finish this today, but I hope that by the time I'm done with this, you'll get a feeling for the complexity of what eukaryotes have to do in regulating gene expression. Eukaryotes are eukaryotic gene expression is really governed by 2 very, very important things. One is differentiation. Different cells in an organism have very different protein needs. The other is that the DNA in eukaryotic cells is not bare naked like it is in prokaryotic cells, but it's wrapped up with histones, and it's very, very complicated structure. Getting the factors into the DNA to facilitate transcription is a much bigger problem than it is in prokaryotic cells. Okay? Well, let's first of all talk about those proteins that are there. If we take eukaryotic DNA and we start peeling apart the chromosomes very, very carefully, what we discover is that they look something like this in an electron micrograph. Okay? What you see is a structure that people describe as beads on a string. And what you're actually seeing is DNA strand wrapped in a coil around cores of proteins. Core of protein, core of protein, core of protein, core of protein. Okay? And these cores of proteins are, they contain proteins called histones. Histones are basic charge, that is, they're positively charged at physiological pH. And DNA is negatively charged at physiological pH. They have a very strong attraction for each other. Okay? And each of these sort of central wrapping things here has a total of 8 proteins, 2 copies each of 4 different proteins. The 4 proteins in there are known as H2A, H2B, H3, and H4. And as I will show you in a little bit, the structure of those proteins very, very similar to each other. Now, there's the beads. The string has DNA, but the string, in some cases, has another histone on it. It doesn't cause the coiling, though. The other histone that we frequently find in the spacer regions is called H1. So there are 5 different histone proteins, for our purposes, 5 different histone proteins, and the region between the beads is frequently occupied by the histone protein known as H1. Okay. The structure of the wound DNA around the 8 proteins on the center creates something called a nucleosomal core particle. That's the most fundamental unit of chromatin. What's chromatin? Chromatin is simply the DNA protein complex. So the most fundamental unit of chromatin is a nucleosomal core particle. This region that you see here is roughly 200 base pairs in length. And you can see that the DNA wraps around it a couple times, and that's what you're seeing in those beads on that string. There's a side view, and that's a schematic. Okay? DNA wrapping here. So we see it's making some wraps around those individual proteins that are in there. Now, this is what those proteins look like. Whoa. We talk about conservation of structure. All right? 4 different proteins all involved in that nucleosomal core particle, and look at their structures. Really, really similar to each other. Structure is important for function. The fact that we see this conservation of structure says this function is pretty darned important. Not only do we see this conservation of structure within an organism, we also see it between organisms. If we were to compare the histone proteins of yeast, one of the simplest eukaryotes, to the histone proteins of human beings, we'd have a hard time telling them apart. So these are really critical proteins. We don't see many changes. We know that function is absolutely essential, and not surprisingly, these guys are having to wrap up those DNAs so they fit inside the cell. As I've said in class before, the total sum of all the DNA of a cell is 7 feet long. That's got to be wrapped up. That is in a human cell, it's 7 feet long. It's got to be wrapped up so it fits. If it doesn't, the cell doesn't have a chance. The cell can't get bigger. Okay. Those individual core particles themselves get arranged with each other, and so we start seeing super helices. Okay? We could have a helix of this, and then we could have a helix of the helices, and so forth. So by the time we see a visible structure in the microscope, like in the form of a chromosome, we're talking about a tremendous amount of packing and ordering of these things together to give that structure that we're able to see in a light microscope. Okay? Well, not surprisingly, okay, if the gene that the cell needs to make is found right here, getting proteins into this, if this is in the middle of a chromosome, is not a trivial thing. The cell has to be able to get proteins in there to start transcription, or proteins in there to replicate DNA or whatever. And so there's a lot of orchestration that has to happen in order to unwrap this structure so that transcription can occur. Now, a lot of what I'm going to be telling you today, or not today, but today and tomorrow and Monday, has to do with the considerations necessary for unwrapping this structure. Transcription can only happen when this structure has been unwrapped. Okay. Well, I talked the other day about some DNA binding proteins, and I said that we saw some common features among those. And the ones I described the other day, the helix, turn helix, are primarily, or are mostly found in prokaryotic cells. Okay. There's some other things that we see in DNA binding proteins, and these tend to be found more in eukaryotic cells, one of which you see on the screen. Okay, so these are protein domains, part of a protein that's binding to a DNA. The DNA is at the bottom, the part of the protein is there, and look at that. All right, there's a beautiful helix that is interacting with the double helix. You see it says it's a basic region, meaning it's fairly positive, and so it's attracted to and it's binding to this DNA molecule. This structure, called a leucine zipper, is really interesting. It's really interesting because when it was discovered, people were very puzzled by it. Okay? The puzzle was when they analyzed the DNA sequence, about every 7 bases, they saw a leucine residue. Leucine, 7 bases later, leucine, 7 bases later, leucine, 7 bases later, leucine, or not 7 bases, 7 amino acids, leucine, 7 amino acids, leucine. Leucine was appearing in a very regular thing. That was kind of odd. Why was leucine doing this? Okay. Well, leucine, you may recall, is a fairly hydrophobic amino acid. It doesn't like water. It likes to associate with other hydrophobic acids. And if you go every 7, the same side it's appearing on. So here, 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 here. Every 7, we're seeing a leucine. That's interesting. Leucine doesn't like water. What if I have another strand over here that has a leucine, 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 leucine? What's happening? The leucines are interacting with each other, very much like the teeth of a zipper do. Okay? So leucine zippers turn out to be a really important structure in not only structures within a protein, but sometimes between proteins. And we commonly see them in features like this, where we have something that, what is that? That binds to a DNA molecule. Okay. Another common structure that we see of proteins that bind in eukaryotes to DNA molecules are called zinc fingers. And they're called zinc fingers because a portion of the protein binds the zinc ion. Oh, what's this thing? I have a backup recorder in case my microphone goes bad, and it's full. Okay. A portion of the protein binds to zinc, and that causes literally a finger of amino acids to stick out. And that finger sticking out is what is the portion that interacts with the DNA double helix. So zinc finger structures are also important structures. If I'm determining the structure of a protein, and I don't know what it does, and I see a zinc finger, I can say, maybe it's a DNA binding protein. Structure can give me some clue to what the protein actually does. Yes, sir?
Are they generally as triplets like that?
Are they generally as, with the orientation here? Yes. In fact, that's a good point. They will commonly have two or three cysteines and a histidine that's helping to coordinate and organize the zinc.
I meant over here with the subunits, you have 3 zinc finger domains all in
Not necessarily that, no. But this you will, yes. Yeah. Okay. Now, I need to introduce another term, a mediator. Okay? A mediator is a person that helps settle arguments, right? A mediator can help make something happen. Cells, eukaryotic cells have mediators, and mediators turn out to be really useful because they can bridge the gap between a transcription factor, and an RNA polymerase. Remember in a eukaryotic cell that the RNA polymerase isn't necessarily, in fact, it isn't the first thing that binds. We have the accessory proteins that bind to the control region, and those accessory proteins ultimately help the RNA polymerase to bind. Right? Well, what a mediator gives is a bridge between a transcription factor that might bind a couple hundred base pairs away and an RNA polymerase that starts 200 base pairs away to start transcription. The mediator will provide, as I said, a bridge between the transcription factor protein and the RNA polymerase. It's a fairly simple concept, but that's what a mediator does. Okay. I talked briefly when I talked about transcription with respect to enhancers. And I said enhancers were sequences that we saw in eukaryotic cells that are very different than things we saw in prokaryotic cells. They're things that are bound by proteins that affect transcription, and they affect transcription in a tissue-specific manner. What does that mean? Well, we see on this particular gene some sequences that are enhancer sequence elements. Let's imagine that in skin cells, I have a protein that recognizes CAG-CTG. Okay? Only skin cells may have that protein. And if that's the case, then in skin cells, that protein is going to bind in this region, and it's going to activate transcription of a gene that skin cells will need. If I go to a muscle cell, it doesn't have CAG-CTG. Well, it's not going to activate there. But if the muscle cell needs it, it might have a protein that recognizes TTAA, TTAA. It might have no proteins that recognize any of these. Right? So if it has no proteins that recognize any of these, this gene won't be made in a target cell, in a muscle cell, for example. So enhancer sequence elements allow the cell to control transcription using specific proteins that are only made in that cell. Very, very useful control system for cells. Very simple. Questions about that? Okay, I said we do one other song. Let's do one other song and we'll get out of here. It's a fun song. I think you know the tune to this one. Everybody likes the Flintstones? All right, let's do histones. Tiny histones wrap up eukaryotic DNA. Using lysine side chains, they arrange a chromatin array. With them, DNAs of 7 feet fit inside the nucleus so sweet. When you use the histones, you have to deal with condensation and its ablation inside your chromosomes. See you Monday. There is. There is. And you saw in that figure that I showed that it didn't just go straight up, that it went up over time. Right. Okay? You bet. Good luck. I'm sorry?
The mediator, is this the same thing as in my genetic stuff? It talks about basal factors sometimes that body will bend it so that
You could consider them in essentially the same.
okay, so it's kind of bending it the same way.
It usually will affect the bending of the DNA, and commonly, and it's not shown in this figure, but commonly these sequences are fairly far apart, and so it actually has to bring them together.
Okay, all right. I just want to make sure I was on the same chart in there.
You got it.
Thanks.
Take care.
I have it.
Yes?
Just a quick question. After we met,